Now I will start with my presentation, which is focused on interaction between hemiparasites and invasive but also expansive plant species. Firstly, let me briefly introduce the problem with biodiversity loss and ecosystem degradation. Maybe I will move here. There are several key drivers which cause biodiversity loss and ecosystem degradation. These 5 drivers were published in a global assessment, and for me and for my work is the most important, the last one, invasion of alien species. Moreover, Milan Chytrý with his colleagues published a red list of habitats of the Czech Republic, and in this publication they also highlighted this problematic native species as very important drivers of degradation of nature habitats in our country. So these 2 groups, problematic native species, so-called expansive species, and invasive species, are, let's say, important actor of my thesis. Invasive species are the bad guys in my story. These species are non-native and which spread during the last year's decade into the new area thanks to the human activities. They have some very important characteristics like rapid and controlled spread, and they are also competitively strong and reproductively very successful. These species also have a negative effect on native biota. They can suppress native species, also form monoculture, and alter entire communities. These 2 species, Solidago giganta and Symphyotrichum lanceatum, are the most important for my presentation. Both of them are from the North America. They are clonal perennial plants with massive impact on the natural habitats here in the Czech Republic. Another important group are expansive species which have similar characteristics and also impact to the native ecosystems, but these species are native, and during the last years and decades they increase their abundances in their natural habitat, but also out of this natural habitat. A nice example is Calamagrostis epigeos, which is a very problematic grass in grassland ecosystems, and also shrubs which encroaching grasslands in Europe, and now it's quite a serious problem across the countries. If we want to save or protect our native habitats, we have to work with these species, and firstly we have to eradicate them and then also restore the native communities. So if we want to suppress them, we have several options. First, the option is that we use standard management methods like frequent mowing or using herbicides. But both of these methods also cause some problems for another species. If we use frequent mowing, then insect will be problem to survive in these habitats. And also these approaches are expensive. Second possibility is biological control. This is a quite common approach in the agriculture, especially in greenhouses or vineyards, and this is based on the interaction between the pest with another organisms, which are their predators, parasites or pathogens. This control can be based on the natural enemy, where we use the specialized species against the invasive species, in our case plant. are from the same origin, so it also can cause some additional problem because of the introduction of new alien species to the new area. Here is the example of the problem. This moth species were used as by control of invasive opuntia, but it's spread it into the native rare cacti in the US. So the effect was more problematic than effectively. Second approach is based on the native species in the invaded area. Here we usually use, or we can think about the species which are generalist with complex of ecological interaction in the community. This is a good possibility, or it works like protection against the invasion and invasion of the new species. And this is a nice opportunity for parasitic plants. In my work, I focused only on one part of parasitic plants, ophiutoid hemiparasites. These plants are green, that parasite on the root system of the another plants. They uptake water and nutrients from the host xylem by the specialized organ haustorium. This is this one, and this is the connection to the host root. This species can reduce host biomass and also fitness. And for our experiment was the most important that these species are mostly generalist, but with some preferences. In the nature, they work as ecosystem engineers. They can reduce dominant species, increase biodiversity, alter nutrient cycling, and so on. What is important that hemiparasites are a natural part of the ecosystems, so don't be afraid of them. They are like a Robin Hood. They take from the rich and help to the poor, let's say, target medium species. In our experiment, we used the native hemiparasites from the grassland habitats, which are unprotected, and for the experiment was important that that this species have enough seeds and easy to collect. So we worked with Serinanthus allophilus, Melampyrum arvense, and Odontites vernus. Let's move to my PhD question. Firstly, we wanted to know if the hemiparasite can form haustoria on various invasive and expansive species. Then we selected some combination of the hemiparasites and invasive species and tried to cultivate it in the field and the cultivation, and tested their suppressive effect on this host species. We also compare two models of hemiparasites and their effects on the invasive plants. And last point was focused on the broader benefits of parasitic plants, which can offer to ecosystems, but also to human society. Let's start with the first paper and with the first question. In this study, we test various combination of the hemiparasites and invasive and expansive host species, and then detect functional haustoria connection via xylem-xylem contact, which you can see here, this connection. For this experiment, we established cultivation pot experiment where we test three hemiparasites, Rhinanthus, Melampyrum, and Odontites, on 18 different invasive and expansive species. In the first year, in the autumn, we planted adult host plant into the pots and sown single hemiparasite seeds into the pots. Following spring, we collect the haustoria from the root system and later we prepared a cross-section and verified xylem contact. We found out that most of tested combination produce functional haustoria. So from this experiment, it looks that the effect of hemiparasite can be quite promising, and based on this table or based on these results, we selected a combination of Rhinanthus aleuterolophus and Melampurum arvense and tested them on the Solidago giganta and Symphyotrichum lanceolatum in the following pot and also field experiment. Here is the second pot experiment. This study was led by Tamara Těšitelová, and the results were published in Neobiota. We prepared the cultivation experiment, and we wanted to know how host identity influence host growth, whether hemiparasites selected in the host monoculture become better adapted, and how these two hemiparasites differ in their effects on the host species. So, in our cultivation, we use Rhinanthus and Melampurum as hemiparasites and tested them on the clonal species Solidago and Symphyotrichum. Firstly, we collect the seeds from the native populations of hosts of hemiparasites, then let's say breed them on the monoculture of one of these hosts, and later we establish this pot experiment. We planted one adult plant of the host into the pot and then sown the seeds of one of the hemiparasites in the different treatment. So the most difficult part. Firstly, we use the seeds of hemiparasites from the same species as the planted plants. So this is called home treatment. Then we use cross treatment, where we use the seeds from Symphyotrichum and sow it to the Solidago giganta. And last one was naive treatment. Here we use the seeds from the native populations. And of course, we also have a control without any hemiparasites, just the host species. And according to the results, Solidago is generally a better host, and Melampurum produce more biomass generally on both species. Yes, here. Than the Rhinanthus. But unfortunately, the effect of hemiparasite adaptation was not so strong. So only the combination of Solidago and Rhinanthus show some effect of this selection in the true treatment, the home and cross. So you can see here, and this two treatment was more suppressive for the host species than the plants of hemiparasites from the naive treatment. For my thesis was the most important, this part of this experiment, where we compare the effect of the both of hemiparasites and also test the suppressive effect of these hemiparasites to the host. So, biomass of the Solidago was reduced by 80% in the pots with the Melampurum and by 50% in the pots with Rhinanthus. And the effects of both of hemiparasites was similar for the pots with the Symphyotrichum, about 30%. So, according to these results, the hemiparasites are very successful in the suppressive effect of both of the species, and maybe Melampurum is a bit stronger than Rhinanthus. Let's move from the pot experiments to the cultivation. We also want to know if the hemiparasites can work outside in the real conditions. So we asked if they can suppress the biomass above ground and also biomass above above ground biomass and also below ground biomass better than standard management mowing, and test whether hemiparasites increase community diversity and support the recovery of the native species. We established multi-site experiment in several localities in the South Moravia with strong before-after control impact design. We use only Melampirum ervense as hemiparasites and tested on the Symphyotrichum and Solidago. During the 4 years of the experiment, we recorded the species cover, height of the invaders, and the last year we also collected the below ground biomass. One experimental block consists of 3 different treatments. First plot was like control treatment without any management, unknown. Second was this mowing 2 times per year, and the last one was the combination of mowing and sowing of the seeds in the first year of this experiment. According to our results, Solidago giganta, the cover of the Solidago giganta was suppressed by 90% in the treatment combining mowing and Melampirum seeds just during the 3 years. And the effect of Melampirum to the Symphyotrichum lanceolatum cover was 70%. Similar or even better effect was for the below ground biomass, where the Melampirum cause biomass degradation by 90%, and in the case with Symphyotrichum, biomass decreased by 80%. Here you can see how it works in the real condition in this photo. This is the treatment among plus Melampirum. This is the situation before we start with management, and this is the last year of the experiment. So I think it's obvious that Melampirum successfully and quite rapidly suppress both of these 2 species. Melampirum also support biodiversity. During the years of the experiment, species richness increased by plus minus 5 species during the per 1 plot, but the, let's say, quality of the species composition was questionable because our plots were in the beginning very degraded. So Melampirum support target value species. You can see here in the green, like Hypericum perfluoratum, Plantago, Lanceolata, Picus heracides, but also ruderal species and grasses, and also some problematic species like Calamabristis epigeos. Last paper was focused on the positive aspects of parasitic plants on the ecosystems and human society. So here we just review the knowledge about this species. In this picture, you can see a summary of the benefits which offer hemiparasites to the ecosystems. They are ecosystems engineers. They can suppress dominant species as you see before, increase biodiversity. They can also create some gaps for new seedlings. support fauna and increase food sources for some animals, birds, pollinators, or herbivores. Parasitic plants are also important for the people as a food source. You can see in the picture Santalum acuminatum. We can use them also in the medicine and perfumes as a drop of good flesh, which consists of euphrasia, or very important part of the perfumes is santal wood. Parasites also have aesthetic value. Here you can see nice modern flower bed with hemiparasitic Castillea. This is the orange one, so this is the future. And they also play important role in the mythology and cultural symbols. And here I have a question for you, and also I have some present for the winner. So, which parasite is the most important symbol, at least in the European context? So, it's a really question, so I wait for the answer. Okay?
Mistletoe.
Yes, mistletoe. Perfect. So this is the present with the mistletoe, also with some aesthetic quality. You can kiss under the And to the end, I would like to thank to all people who helped me during my research, and also thank you for your attention.